Hello and welcome to another episode of the Computinomics podcast. Our guest today is a member of CEPLAS, aka the Cluster of Excellence on Plant Sciences, and he's a group leader at the Institute for Plant Sciences at the University of Cologne. Markus Stetter, welcome to the podcast.
Hello, thank you.
Markus, we like to start with a little bit of an icebreaker, and I was wondering, what's your favorite Sunday breakfast choice?
Oh, it's very diverse actually, but usually it's different pastries, mostly French pastry, to be honest. And so, yes.
Or should I say, ooh la la?
Yeah, probably.
It sounds delicious.
As during the week, I eat only cereal, so on the weekend, I treat myself.
Ah, nice. But that's actually, I mean, the cereal is kind of why I asked this question. So my ulterior motive was a little bit of a reference to your research because you study the domestication of Amaranth, which many people, including myself, know mainly as a breakfast cereal. So can you tell us what exactly are you focusing on in that research?
Mm-hmm. Yeah, so as you say, you mostly know it from breakfast cereal, but actually it has a very exciting history as a plant and as a crop. Because when you eat it in your cereal bowl in the morning, what you would not know is that there is actually 3 different species of grain amaranth that have been domesticated. And this in different regions of the Americas, 2 in Central America and 1 in South America. And this is sort of very interesting for us because it gives us a replicated experiment that happened basically 3 times and over about 8,000 years. So to replicate this, we would need 8,000 years. And here we have this setup of an experiment right in front of us. And there's typical changes that happen during domestication. And some of these we see in Amaranth, but many actually not, and that makes it quite special as well. And this is one of the reasons why we study this pseudo cereal actually, and we eat it in our breakfast cereals, but it's actually not a cereal because that would be only grasses.
Mm-hmm. Let me follow up. You said there are some maybe typical traits and some that are less typical or that are maybe special. First, the typical ones that you focus on, what are they?
So we compare this with other crops, and in our sort of our major crops, particularly our cereals, changes that we really see consistently, independent of where they were domesticated, is sort of an increased seed size, or the fact that the seeds don't fall down anymore, so that they don't shatter anymore. And this has simple reasons of this human cultivation, where humans want to come once and take everything home, right? So for the plant, is sort of an incentive to have more seeds that stay on the plant. For a wild plant, that is not good. In our Amaranth system, we don't see those typical changes. The seeds are still very small. They have a small increase compared to their wild relatives and wild ancestors, but not very much. And they still fall down relatively strong. And even though this happened 3 times, and we know from sort of historical and archaeological evidence that people have used it actually over that time. And in some periods of time, they were as important as major crops like maize and beans. Yet these changes have not happened. And so I'm particularly interested in why certain changes did not happen, which is the hardest to look for, because if you don't see them, you don't know, do you not see them because you're not good enough at looking, or are they actually not there? So we start by looking at some that really did change. And there are such changes, like actually these 3 times. One very obvious one that we study, and because we can study it quite nicely, is a change in seed color. So all your breakfast cereal amaranth that is mostly puffed, but it actually has this white thin seed coat, which makes it, which comes from a pigment that is accumulated in the seed. And this pigment seems to have gotten lost 3 times. And there we can molecularly dissect even what pigment this is, what genes make this pigment, and why this pigment was actually selected against during domestication is some of our research that we're trying to study. And so to better understand how does a plant how do we make new crops, right? Or how were crops actually, made? They were not made, right? But how did crops become crops? And how do plants in general adapt to change? And here the humans made the change, but again, humans are making change for the future differently this time, and we want to understand how this works in order to better predict how adaptation works.
Mm-hmm. And how do you go about tracking these changes? So you are looking at 3 different types, but what methodologies do you use to figure out how these changes happened, and hopefully get a few ideas of how we will be able to make them happen?
Mm-hmm. Yeah. So here, and this is the other sort of nice system about this domestication gradient. So because this happened in such a short time, in only a couple of thousand years, which in evolutionary time span is very short, the local relatives and ancestors basically still live in the native range of where those plants come from. So here in the Americas. So we, for one, can compare the 3 crop species to each other. But what we mostly do is we compare them to their wild ancestor. And here comes the other special sort of for Amaranth, at least what our research shows so far, is that they have been domesticated 3 times from the same ancestor. So we really have this replicate. But this ancestor is spread all around the Americas. And so what we do sort of methodologically, we sequence hundreds of genomes of these wild ancestors that were collected along the Americas, and such Amaranth land races, some more, some less cultivated. There's not very many bred ones, but there are some that were sort of improved and come out of early public improvement systems, and we compare those. And later, just like other crops as well, this crop also spread around the world. So nowadays, actually, the Americas is not the biggest producer anymore of Amaranth. So we also look how this spread sort of worked and why it worked, and where this is being grown now. So so we use a lot of population genetics as we have worked with this natural diversity present in the genomes. But then when we look at the molecules, of course, there goes metabolomics and transcriptomics to understand, how are genes regulated, which genes regulate which metabolite, and therefore a certain phenotypic trait afterwards.
Mm-hmm. And you've referenced that what you want to find out, like how and why the spread worked the way it did. Do you have any results that you can share already of this part of your research?
Yeah. So the first thing, what was not really known before we started this, is which wild species, because there's about 70 Amaranthus species, or up to 70 Amaranthus species, that some of them would be potential ancestors. So we found this ancestral species, and our population genetic analysis showed that this one species is most likely the ancestor of all 3 of them. But in a very recent, or at the moment, a preprint manuscript, we find that although these were domesticated in very different regions, there's still gene flow. So in basically long distance exchange of genetic material between those crops after they were domesticated. So probably hinting at human exchange over long ranges, so even between South and Central America. So this was independent, but yet there was some interaction afterwards again. And so this is sort of on the bigger picture on how did this work on this metabolite or pigment change. We have identified very promising genes actually that we are now molecularly dissecting and cloning that gene to show what it does. And actually we are, I think, very close to, at least in one species, to find the actual mutation that occurred during this selection process that led to this change. And interestingly, this seems also to be not the same one in the three species. So different mutations, but in the same gene, leading to this change.
To this pigment change.
Exactly. Yes. Yeah. And the pigment change itself sort of has an ecological meaning that we're sort of working on. Yeah. This is still very fresh work there. So yeah, but this is what we're trying to figure out.
You're anticipating a follow-up question here, because I was just about to ask.
Yeah, exactly.
If there's some conclusions you can draw as far as the ecological impact.
Yeah. So I mean, most people often think, and I mean, for a long time we sort of, I mean, it is a compelling question to just think, well, maybe it was just a curiosity by people that these pale white seeds looked very different to all those to those black and dark brown seeds, and it was just a curiosity. But that seems to be not the only case. There seems to be really ecological reasons why a crop is better without those pigments than with those pigments. And this is sort of what we're currently trying to figure out and understand experimentally as well.
Interesting. You've, I think it was a 2021 interview with a local Cologne paper, you described your work as basically fast tracking evolution. So this is the work you just described would be exactly that, right? You're trying to fast track evolution and try and understand how it happened and how we can apply it ideally to crops today.
Yeah, so we're doing this on two levels, and this is already fast, right? Evolutionary, this is very fast. This is 8 to 10,000 years or so, but we're even looking in sort of shorter time spans. So what happened once we have a crop, right, which is already adapted to this human and made environments and can live the way we want crops to live, right? Then those crops often spread all around the world. And this, of course, only started to happen once the world was sort of globalized and connected. So in the last 500 years or so. And there, crops have done this very amazing job of actually spreading everywhere. And here we do this for one, I said for Amaranth, where mostly we study how it came to India, for example, because there it's now the biggest, India is the biggest producer of amaranth in the world nowadays. So how it got there, and it only got there once Europeans spread all around the world and spread also those crops. And we do this in comparison with this very well domesticated plant with maize and how it came to Europe. And there we study sort of how this happens in this very short time, so only 400 years, because this is sort of one of the important evolutionary questions looking forward, thinking of things like climate change or larger populations, is we might not have 10,000 years, right? So can populations, and particularly crops, which have had this strong pressure of selection already to become a crop, do they still have the potential to further adapt, and how fast can this go? And how does this go? So does this go fast is what we want to know. And if it goes fast, why and how can we maybe even make it for certain ones go faster, or for others we might not have to help, and for some we might have to help more. And yeah, so this is sort of the fast track evolution that we're trying to look at here.
Right. You described a few results on the other aspects of your research. Do you have similar kind of intermediate results with regard to these questions? So an answer to, okay, how can we make plants adapt quicker in a totally different geographic setting?
Yeah, I think the major result is really, a lot of it is about diversity and how much sort of standing diversity we're starting with, right? So how much can we bring in from other relatives? We see this in our work of the amaranth in India, that the three species actually got back together in India and sort of shaped new populations by interbreeding there. This is sort of the preliminary work, what it shows. In maize in Europe, we see also that although there is sort of locally forming populations, there seems to be still quite some exchange, but actually in some of those very local land races and populations, this seems to be more the difficulty. So their sort of assisted exchange, of course, would be beneficial if we want to further breed or improve those crops. So diversity plays an important role there, and what is important for us as well to transfer sort of these ideas and this knowledge to sort of other population systems as well, right? We can think of that for a natural population, this would be the same, right? If there's a very local species that grows only in a certain area, it might be important that it has still connection to other populations of that wild species in order to be able to adapt to things like climate change. And so if we understand this better, we also think that this will be important for conservation of plants more in general, but of course of crops in particular.
Right. Makes sense. I mentioned in the beginning in your introduction that you and your lab are a member of CEPLAS, the Cluster of Excellence on Plant Sciences. Can you give us a quick rundown? What is CEPLAS?
Yes. CEPLAS is, as you said, this cluster of excellence, one out of 57, I believe. And the main one working on plant sciences, and under the sort of slogan "Smart Plants for Tomorrow's Needs," we do this. So we try to understand fundamental processes in plants in order to understand how plants work and how they work together. This we do sort of in sort of a fluid structure of 4 research areas. One working on plant microbe interactions, so we don't look at the plant alone, but with its interactors. One, we try to optimize plant performance by understanding how development and metabolism work together. Then one where we look at synthetic and reconstruction biology, so how can we make those processes artificially, and one on theoretical biology and data science, which of course is very important nowadays. And all these sort of fluidly interact by now since the first phase. And maybe very important is to say that this is not just us at the University of Cologne, but it's a joint cluster between the University of Cologne, the HHU in Düsseldorf, the Max Planck Institute for Plant Breeding Research, and the Forschungszentrum Jülich. So we all got together, and it's over, I think, over 50 different labs involved and several hundred scientists actually working together on this major goal of understanding how plants work and how we can sort of work on the plant of the future.
Right, the smart plant of the future.
The smart plant of the future, exactly.
For those who haven't like maybe an idea or who think of what's a smart plant? Is that a computer and a plant? What does it mean? How would you define what would be a smart plant, the ideal plant that comes out of your research that you help design?
Well, yeah, I think that there's a lot of many layers to it, right? This is where we try to understand how these different processes work from the development. So how does two different cells get together to make a certain tissue? How do metabolites get transported through the plant, and how does this interact with the environment? And so the sort of smart plant of the future, which, to be honest, we are of course far away still, but we aspire to get to this, right? Is the plant that sort of is able to react on its surroundings and sort of integrates all these things. Of course, plants do this, right? But we want to sometimes, especially for crops, we want the plants to do this in a way that we can predict. And so, in some parts, we try to design certain parts of this of such processes. But the idea is not really designing this, but to understand how these plants are so smart and how they can actually do what they do.
Right, which in a way would form the basis for designing and at some point, hopefully, breeding these plants. So we have them
Exactly.
to consume, to feed the many more people that we will have in 2050 and further on, right? One thing that also struck me about CEPLAS that I saw on your website is that you define not just a research mission, but basically also a training mission, going into novel forms of training for undergraduate and graduate researchers, postdoctoral scientists. What is that? What are those novel forms?
I think CEPLAS has really over the last years become very strong and very innovative, where we're trying to sort of pick up the researchers very early on. We start with their own undergraduate program in quantitative biology, where we really train already starting from after the bachelor's with computational mathematics, together with biology, because all this involves biology, and sort of along the evolutionary process, also conceptually different than maybe most of us have been used to study biology. So we have this undergraduate program, and then for our graduate school within CEPLAS, so our PhD students, we actually pick them up after a bachelor's degree. So in difference to the common system in Germany, where usually students do a master's first and then go into their PhD, we have sort of this fast track PhD program where the PhD students have a 1-year training phase where they sort of rotate through different labs, but of course get also the theoretical foundations for their work. After this first year, or towards the end of this first year, they do 3 6-week rotations in different groups within CEPLAS. And then the students actually get to pick the lab where they want to do their PhD out of those. And this gets or pick or it gets matched basically. And so they can really find what they are interested in within the frameworks of CEPLAS. And then they have 3 to 4 years to complete their PhD within this. So this is sort of the basic training, but we don't stop there yet. We continue for postdocs. We have a postdoc group that is jointly recruited, just like the PhD students. Actually, maybe this is also a very strong difference to the general German system, because they come in a cohort, so they have sort of this community support of people that started at the same time. We do this same for our postdocs, which come in within this postdoc training program. They also fit into the different labs and have this cohort feeling, which is combined with soft skill training or beyond. We have interaction with industry, interaction with other scientists. And then there's also even mentoring programs for people like me, young group leaders, where we even have a peer mentoring group where different group leaders, even from different fields, other Max Planck institutes for aging, for example, and other clusters of excellence, get together, exchange experience, and sort of get this sense of community as well that is really lived throughout CEPLAS, that there is a bigger thing than just the sum of each group. And then we even have outreach programs actually starting from high school students and before up to seniors, which we give lectures on different plant science topics. So
so outreach in the sense of scientific communication, public engagement, rather than trying to get the young minds into that field, or both.
Well, to some degree, both, right? We have this children's university program where really I think it's even primary school children that come for a day into the university and can see how cool plants are, right? Because often when we think about biology, actually, we forget how cool plants actually are, and often get showed other things. Here, we really show them plant sciences very early on to excite them for our topics. And then, of course, outreach around this is also a more and more important factor. But really, all those levels we try to to work on education on plants and sort of bring the next generation of plant scientists along with us.
That's pretty cool. Last follow-up, you mentioned, and I think that's really exciting aspect of your training structure, which seems to be like very overarching, is that idea of helping not just kind of the early stage researchers, but also group leaders, mentorship. How much have you taken advantage of this, or maybe what's something that you've taken away from this mentorship structure?
Yeah, so this group leader program. So this is particularly for me, being a group leader at a university where those are sort of less common. In the Max Planck Institutes, for example, those are a bit more common. So it's really nice to speak to my colleagues there, right, and be able to exchange and say, oh, how do you do this? How do you do this? See also what problems people have. They are often very similar, right? To get started. It's particularly for group leaders within the first couple of years, and so this is really really nice to have this community and be able to exchange, but very hands-on. CEPLAS is also supporting this sort of with funding. So we as group leaders have full access to the graduate school, can write proposals for this. So I have in my group 2 PhD students that are funded through the CEPLAS graduate school. And other support from CEPLAS beyond, also with the of course with the more established groups, and there is really a nice exchange, not hierarchical or anything, but everybody is very nicely working together, and this is really what I enjoy in being part of CEPLAS.
Very cool, Markus. Thank you for taking the time, letting us in to your research, your work with CEPLAS and the consortium as a whole. I hope to maybe have you back in a year or 2 years' time, so you can share more of the results that you have. For now, just wanting to say thank you, and keep enjoying how cool plants are, and that amazing training structure.
Thank you very much. Thank you for asking questions and being interested. Thank you.
My pleasure. And to our listeners out there, we hope you enjoyed this episode. If you want to learn more about Markus's work on the domestication of amaranth and maize, about CEPLAS, head on over to computomics.com. Our show notes will include links to Markus's lab page as well as CEPLAS. And as always, feel free to check out previous episodes as well. We look forward to having you back next time on the Computomics podcasts.